I'm going to tell you some of our experiences within trade, with what we did with the Postgres version of tranSMART. We, as trade, adopt a process where we, in somewhat larger steps, so 3 steps a year approximately, take from different studies that we want to support use cases that we try to realize and support in tranSMART. So this is a first use case that we started with a few months ago. It's a use case on copy number variations. It's from the Cairo study, and the study in itself is a colorectal cancer patient study where they investigate possible relations between chromosomal aberrations in patients with metastasis and their response to treatment. So in those studies, a lot of data is, of course, collected, clinical data, omics data, non-omics data, and genomics data. So gender, of course, DNA staging, age, non-omics data like microsatellite stability or instability, whether some of the genes have been mutated or not, so the KRAS or the BRAF. And genomics data is the comparative genomic hybridization microarray data that is used in the study. Something about what it is, although I'm not an expert in that, but I hope I can tell you, explain a little bit what it is about. Chromosomal aberrations are a key event in the development and the progression of cancer, and the array-CGH is a high-resolution method to detect chromosomal DNA copy number variations. So normal tissue, normal somatic cells have 2 copies of the chromosome. There are, of course, the normal male cells only have 1 copy of X and 1 of Y. In cancer tissue, the copy number may vary considerably. So the method that is used is labeling 2 samples of tumor cells and non-tumor cells, normal cells, with different labels. Here in colors, red and green. They are hybridized to the microarray, and in this case, a microarray platform used with 180,000 probes on it. Depending on whether there is an excess of tumoral DNA or a lack of tumoral DNA, or it is comparable between the tumor cells and the normal cells, you get a different coloring of the microarray. That needs to be analyzed. So the image is taken, I guess, and it is analyzed for further processing. So this is what comes out of when you have the data. It shows you the log2 intensities of what is measured across the genome, so at the different chromosomes. Of course, a number of processing steps the data will undergo. In this step, what is called segmenting, you try to find the areas where the copy number variation is similar. So you instead of looking at the probe level, you try to extend that to areas where it is similar. Statistics metrics are of course behind that. And the next step is that you try to come to some conclusion whether there is a significant raise or a loss based on the probabilities you define and a threshold you define whether some of the observations are a gain, so an excess of tumoral DNA in those samples at those locations, or a loss, or whether it is normal. Then there is another step that tries to combine all the results for all the patients that you had in your group. Those segments differ from patient to patient, so the breakpoints between the segments are not the same for all patients. You try to harmonize that, so you come to intersections, and you come to what they call chromosomal regions, where you have your call, either a gain, a loss, or an amplification, or normal. At the chromosomal region level, as we say. So it's not anymore at the probe level, so you have much less. In our case, it was, in this example, 9,000 chromosomal regions, where you look at instead of 180,000 probes where you have information. I think there's a note on generating it. So what can you do with the data? Of course, you can analyze the data. We consider two main analyses in this use case. One of it is a group test, so you compare different groups of patients, compare their chromosomal aberrations, have a look at can we spot any differences in that. So in this example, you look, for example, at gains and losses, the frequency plot of the gains and losses for patients with microstability, have a microsatellite stability, and here the microsatellite unstable patient group. And another type of analysis is survival analysis. You can, of course, also look, if you have survival data of those patients, compare at each chromosomal level whether the groups of loss, for example, versus no loss patients that have a certain chromosomal position or loss, compared to a group that have no loss at that chromosomal position or chromosomal region, whether there is a significant difference in survival rate. And you can do that for all the regions that you have. Okay, so this process we try to support in tranSMART. So we made some changes to tranSMART. We introduced some database tables to hold this specific array CGH call data and the chromosomal region data. We adapted more or less, created an ETL pipeline for uploading that data. It's very similar to the gene expression pipeline. So we basically started with that pipeline and adapted it. We encapsulated a number of scripts, R-based scripts, to do the analysis for us. And we created some of the UI elements that are needed to be able to specify the inputs for running the analysis. So they will appear in the advanced analysis tab as new analysis items. Yeah, this is maybe for the other group. Some of the details of the data database tables, but the only thing I want to take out here, that in fact there are this table and this table are new tables, but they look also in their names very much to the tables that have holds similar data for the microarray for the gene expression data. Okay, okay, we will see that in the demo, because I think I want to switch to the demo now. Okay, why of tranSMART. So we have here a number of studies. I will focus on this colorectal cancer study. Let's just select the whole study for the time being. I will not go to some of the exercises that Johannes also already did, because you can also do here the exercises with the clinical data, so to say, with looking at different groups, age distributions, but also other types of distributions on survival and so on. Of course, I could, let's see, let's do a little exercise to see. So this is what we have uploaded. In here we have the data, the array CGH data as a high-dimensional data. We have also some other observations with respect to mutations of certain genes. We have data about survival times. We have also data about the microstability of the microsatellite stability. And for example, yeah, you can of course make two groups out of it, generate statistics. You can have the statistics. You can also, so we have the endpoint. So we're looking at survival time. We can then have a look at the distribution of the survival time of these different groups, or we also have the progression-free survival time. So it's another thing you can have a look at. In that way, you can of course investigate your cohorts, different patient groups. So what we you could of course do from the advanced workflows do survival analysis. We have survival times. Run a survival analysis. Have a feeling about the survival analysis of the whole set. You can do the same thing for if you compare the two groups, the MSI and the MSS. You can compare those groups for survival analysis. That's, I think, okay. I expected a different picture, so I go to another thing. So I now want to clear. I want to select all the patients in Cairo 2 and show you some of the tests we included. So here is a group test, as I explained, for the array CGH. For that we need to specify the array CGH data, and again, of course, the groups that you want to compare. It's similar as in the other things. There are some options for the algorithm, and I have to specify how to build the group. So I can specify to compare the gains against the no gain, or loss against no loss, or to specify more groups. So I will select the gain and start the analysis. Now this analysis runs for a while because there's a lot of statistics involved, and to get also some of the nice statistical results, a lot of what is called permutations have to be done to get some meaningful statistic results. So that's why we chosen to be able to put these jobs into a background, and we can later on refer to results that have been put in the background and have a look at it at a later time. Let's see. Okay, good. So not all have completed or are correct, but once so here you have the comparison between the, maybe it's a little bit small, of course. So here you have the frequency plots of the group test. You have the comparison. In this case, it was the groups were whether the two groups were the BRAF mutation group and the wild type BRAF wild type group. Also, we could specify, say, the survival time analysis for array CGH. I was wondering whether, because we can, for the survival time, the same problem applies, that it takes quite a couple of hours to run to get some decent results.
Is it 2 hour something? Is it something unusual? If you run it an hour, would it still take as long?
Yeah. It doesn't have to do anything with tranSMART itself. It's only the R, and still, if you optimize the machine with for performance and have multiple, still it takes a long time. Yeah, but it depends on the accuracy you want to have with respect to p-values, fault discovery rates, and so on. So it's statistical.
There are chance to also look at the data for one particular section of this plot?
No, we cannot. No, we cannot. There's not an interactive browsing capability. So it's an image. You can enlarge it a little bit, but there's in this browser it's.
So I probably have to get the R data again and plug it into some other tool in order to go in depth analysis.
If you want to zoom in or we now just did it with images, but you can of course also try to include a genome browser or what.
See a particular spot of interest, I would like to go in and see whether this actually corresponds to some sort of genomic or something.
Yeah, but so currently we didn't do that, but we are discussing whether to include also in tranSMART some capabilities for browsing genomes for patients.
Should we add to the parking lot?
Internal versus external.
Sorry?
What was the parking lot?
Oh, the browsing, genome browsing, that you can have a closer look at.
Make it generic in the sense of queuing related with the raw data. Yeah. And then specifically ITP or browsing.
Put zoom in so we just get and understand what we can get directly.
So I am not able to find an analysis that for which the data is still available here. So I'm sorry for that. Janneke, can you help me? No.
So now you've added the capability of ACGH, can you select patients based on regions? In your initial subsetting, can you
That not yet, but so that's the thing that my neighbor often asks me to have. So he wants to select a patient cohort, the patients that have a 20Q gain. And that's indeed something that that is not possible. So now you can only also with the ACGH you can only select the high dimensional data as a blob more or less. But if you want to subset that, so you want to specify, okay, I only am interested in not all the observed values, but only in chromosome 20, the Q arm, and I'm only interested in patients that have in that part of the chromosome somewhere a gain, that you would be able, that you cannot specify. But if you look at the microarrays and gene expression, the things are there. So I think you should be able to harmonize that so that you can specify that. Yeah.
What about the linkage between region chromosome and genes that are in that region? Is there a way to connect annotations about that region so that you can say, okay, I'm only interested in gains in or losses in P10?
Not implemented yet. So if you looked at the data tables, you made a picture of it. You can check. I think there we also accommodated for not only the cytoband information but also the possibility to annotate the region with the genes that are related to that.
That seems to be that's going to be part of the kind of core reference functionality that should just be able to connect to that reference data set, wherever it is. I mean, is that there already? I mean, in the core reference data, is the reference data for the human genome?
No, it's not. It's
It's not there. So, for example, for the gene expression data sets, microarrays, if I'm picking genes, how does it know which probes belong to which genes? Is that all specified in the data set itself?
no. That's specified in what's called the platform.
Yeah.
So
It's
the platform annotation.
Yeah. For each platform that you use within tranSMART, at least for the gene expression, not for this stuff.
Yeah, this also has a
annotation. So the entire annotation field that you probably get from AFI or from Geo, from any platform that
So
you're
you just
using.
have to add that once.
Yes, once, and then you search for in the ETL procedure. You actually ask it to see does it have information in this platform, and it tells you it does or it doesn't.
But here is also a platform, but it's what we call the chromosomal platform. The tables are there, but in this case, the platform changes from patient group to patient group, because the chromosomal regions are determined based on the breakpoints that you find in each individual subject.
Probably a bit much to ask from the clinical variables, but wouldn't it be possible to store things like this patient has a Q22 amplification as a clinical variable and then select the set?
Yeah, that would be possible. Yeah. Yeah, that's what you saw in the KRAS mutation. That's a clinical observation derived from an omics observation, but it is handled as a simple observation and not as an omics observation. Yeah.
To some extent, I can imagine we need to express things at the nucleotide level, because you have these questions on the genes that are expressed, you have them on the array CGH on regions that are gained and lost. You will have them in the GWAS studies on nucleotides that are polymorphic. You may have them in proteomics once people decide on phosphorylation sites. You will have them on the splice variants that come up. So somehow I think the question whether to make a choice on what is your basic way of looking at the genome and getting your data connected to that is quite critical.
Mm-hmm.
You should basically be able to transport some of the genomic features you find on the way into a clinical sort of variable setting that you say, okay, I all of a sudden I found a P10 mutation. Let's move this to as a clinical variable into the sample description. I found the gain on wherever.
Sure, but then P10 should be defined as this is a gene in chromosome 10 from this nucleotide to that nucleotide, right?
Yeah.
I don't know how it works, but somehow, I mean, that's one question, and you want to search for P10 because that's the gene that you know. But later on, you may want to search for phosphorylation sites as well. You want to search for splice variants, you want to search for etc. So somehow there needs to be these reference lists of how you will connect the queries that you want to have with the genome information.
Okay, but would that be something that you would consider for an ontology server, for example? That kind of information you'd be able to find lists, or at least all the annotations related to the genomic information at least. When I'm talking about the clinical